Okay, so we're here on the outside of your plant, and we're going to focus in on the outer bark, which is the periderm. And the periderm consists of several layers. It's going to be bound by the epidermis on the outside and the cortex on the inside. So the cortex are those large cells, kind of clear because their contents have been removed through the process of the tissue preparation, but these would all be your parenchyma cells again. And then between the cortex and the epidermis is our periderm. So we have the phellum on the outside of our periderm. Then we have the phellogen. And then we have our phelloderm towards the inside and touching our cortex. The epidermis is usually going to be a clear layer of cells on the outside if you have a fairly young stem or trunk, but it gets split and kind of raggedy and comes off during growth, and so sometimes you'll see it on the outside of your stem and sometimes you won't. And as I mentioned, usually it is a clear layer of cells. The phellum is also known as cork, and that's going to be fairly darkly stained because of all of the chemicals that are put into these cells. And you can imagine if this is your first line of defense, once your epidermis kind of crackles from growth, that you would want it to be a nice barrier against everything you could think of. And so plants put a lot of secondary chemicals into their phellum, into their cork, to help fight against attacks. So bacteria, viruses, fungal pathogens. It also is a barrier to water, so that water is just not constantly leaking out of the trunk. So you'll see a pretty dense, heavy layer of cells forming the phellum. The other area I want to talk about next is actually the phelloderm, and the phelloderm is much larger in size, not as filled with those waxy substances like suberin, and you can see in the image that they actually still have their nucleus. So they're still living, whereas the phellum will function even after death because of everything it's filled with. So that phelloderm again is just going to run up against the cortex. And then with your between the two layers, sometimes it's difficult to spot, but it is often slightly different in color or slightly different sizes. We're going to have another region of cambium, And this is the cork cambium, and here we're using the botanical term phylogen for that layer. It's usually 1 to 2 cell layers thick, so it's quite thin, and it's easier, I think, usually to pick out the phylum and the phyloderm and then just realize that the phylogen is between those layers. And just to reiterate what we've drawn here, anything outside the vascular cambium, so that would be way over here, it's not really in our image. So all of that would be bark, and then here from the cortex out, we have our outer bark. So I think that completes our drawing of the periderm.